Good afternoon. I'm Lorraine Rosamelia. I'm a staff dermatologist at the Geisinger Health System, practicing in State College, Pennsylvania, and I'm part of the education committee for the American Acne and Rosacea Society. So we were part of choosing the abstracts that would be selected for today's events. I'm excited to moderate the second half of the session because it's our younger set, our promising scholars. And so the next is going to be Stephanie Kao. She's a graduate student in Jenny Kim's lab at UCLA. And she'll be talking about Pentobra, which is a novel antimicrobial compound with lytic activity against P. acnes. Welcome.
Oh wow, Dr. Kirby was right. This room is very, very large. Well, thank you AARS for inviting me to speak today. I'm very happy to be at SID and also in Atlanta. Today I'd like to talk to you about Pentobra, a novel antimicrobial compound with lytic activity against P. acnes. So as you all know, P. acnes is a commensal human bacterium that is implicated in acne vulgaris, and increasing levels of P. acnes resistance to antibiotics highlights the need for new therapeutics. In fact, the incidence of P. acnes antibiotic resistance has increased in the past 30 years. Quite simply, the antibiotics that are being used clinically, i.e. tetracyclines and clindamycins, just aren't as effective as they used to be. So we sought to see if another antimicrobial, an aminoglycoside, could work. In this project, we wanted to generate a novel antibiotic against P. acnes that could evade existing resistances. However, aminoglycosides have limited effects on anaerobic bacteria like P. acnes because of reduced uptake. Typically, aminoglycosides are very potent antimicrobials, but they're just too big to be internalized into the P. acnes bacterium. So what we wanted to do it's hypothesized that P. acnes' intrinsic resistance against the aminoglycoside stems from a low proton motive force across the bacterial cell membrane that inhibits antibiotic internalization. So therefore, there exists a need to generate a novel molecule that can self-promote uptake into bacterial cells. So our aim was we sought to engineer a novel P. acnes antibiotic by combining the potent ribosomal activity of aminoglycosides with the robust membrane permeabilizing abilities of an antimicrobial peptide. So Pentobra is a two-part hybrid antibiotic composed of tobramycin, which is a big, bulky, hydrophobic aminoglycoside conjugated to a 12 amino acid peptide sequence that we'll call pentpeptide. The way it works is that the cationic pentpeptide is attracted to the largely anionic peptidoglycan of the bacterial cell wall. From this electrostatic interaction, the pentpeptide is able to poke holes and permeabilize the cell membrane. Tobramycin slips in, targets the bacterial ribosome, and forces mistranslation, inhibition, and eventually cell death. So first we wanted to test Pentobra's bactericidal activity against the P. acnes lab strain. We conducted CFU assays, and what we can see is that Pentobra is highly bactericidal against the lab strain, causing a greater than 5 log reduction in the number of viable colonies as compared to pentpeptide and tobramycin, which were not as highly as effective. So from here we demonstrate that Pentobra is bactericidal against the P. acnes lab strain. After demonstrating that Pentobra was bactericidal against the P. acnes lab strain, we wanted to test if our antibiotic was clinically relevant. So we expanded our study to include more P. acnes clinical isolates that have been phylotyped, ribotyped, and associated with different states of the acne disease. And these are the additional P. acnes clinical isolates that we've used in this study. So we conducted more CFU assays using the expanded number of clinical isolates, and what we found is that Pentobra is bactericidal against most of the P. acnes strains that we tested. So from here we determined that Pentobra is bactericidal against a broad range of P. acnes strains. We also conducted transmission electron microscopy studies to image how Pentobra acts on the P. acnes cell. So in the upper left, we see a normal, untreated P. acnes cell. Note its normal pleomorphic structure and, I guess, general happiness because it's not in any mortal danger of being attacked. And then in the lower left, we treated the P. acnes with pentpeptide, which, if you remember, is that cationic antimicrobial peptide that can poke holes and permeabilize the cell membrane. And consistent with that idea, we see perforations in the cell membrane right here. In the lower right, we treated P. acnes with tobramycin, which is that big, bulky, hydrophobic aminoglycoside that can internalize into the P. acnes cell. So consistent with that, we see that the P. acnes cell treated with tobramycin is nearly indistinguishable from the untreated P. acnes control. And in the upper right, we treated P. acnes with our hybrid antibiotic drug, Pentobra, which combines the membrane permeabilizing activity of the antimicrobial peptide and also the ribosomal activity of the aminoglycoside, tobramycin. So what we see is blebbing and cell membrane permeabilization and also externalized cytoplasmic contents. So we wanted to test if Pentobra could be used in a clinical environment. So we isolated microcomedones from acne patients at the UCLA clinic, and what we found, and we conducted CFU assays on them, and these are representative graphs of our donors. What we found, again, is that Pentobra is highly bactericidal against the P. acnes in the microcomedone as compared to Pentobra and tobramycin, which weren't as highly as effective. What's important about this slide is that Pentobra can be used in a sebum-rich environment in which P. acnes is found in the acne vulgaris disease. So inflammation induced by P. acnes plays a key role in acne pathogenesis. Therefore, we sought to see if Pentobra has an immunomodulatory effect on the host's innate immune response. We isolated and co-treated human keratinocytes with P. acnes in the presence of Pentobra and tobramycin, and also conducted real-time PCR studies to measure gene expression of specific chemokines. So here we see that P. acnes upregulates MCP1 expression, and Pentobra is able to downregulate that expression by about 80%, and tobramycin is able to downregulate that expression by about 50%. Here we see that P. acnes is able to upregulate CCL22 expression, and Pentobra is able to downregulate that expression by about 60%. And here we see that P. acnes is able to upregulate IP10 expression, and Pentobra is able to downregulate that expression by nearly 100%. And here we see that P. acnes is able to induce IL-12P40 expression, and that Pentobra is able to downregulate that expression by about 40%. So from all this data, we determined that Pentobra and tobramycin modulate specific chemokines induced by P. acnes. Lastly, we wanted to test Pentobra's toxicity against human cells, so we incubated PBMCs, keratinocytes, and sebocytes with Pentobra for 3 days and measured the human cell viability. What we see is that the Pentobra demonstrates no adverse effects on human cell viability. And what's important is that we determined that Pentobra is non-toxic and can be applied in real life. So in sum, increasing antibiotic resistance is a problem. The incidence of P. acnes antibiotic resistance has increased in the past 30 years, and the antibiotics that we're prescribing clinically just aren't working as well as they used to. So we sought to create this new drug called Pentobra. And what we found is that Pentobra drastically outperformed free tobramycin, which generally displayed weak activity against multiple P. acnes strains. Electron microscopy studies showed Pentobra has robust membrane activity. Antimicrobial activity was maintained within comedone extracts isolated from acne patients. And our big takeaway message is that equipping aminoglycosides with an additional mechanism of action of membrane activity is a viable approach for targeting anaerobic bacteria such as P. acnes. So we'd like to thank Nathan Schmidt and Gerard Wong in the Department of Bioengineering, Andre Costco for imaging, and members of the Jenny Kim Lab. Thank you.
The questions?
Yes.
Do you have any clinical data?
Do we have any clinical data? No, not yet, sorry.
Hi, I was wondering how you did your assay when you took the lesions from acne patients and incubated it with their drug, and was it just in culture you added the drug?
Yes.
And on that note, how do you think that this drug might be implemented vehicle-wise? Would it be a oral tablet or a cream or?
I think we might be going the topical route.
Cool. Thank you.
You're welcome.
Hi, very nice talk. Do you have any evidence that P. acnes will not develop resistance to this new form of tobramycin?
That is a very good question. So aminoglycosides are typically potent antimicrobials. For example, there's the streptomycin that was initially used against tuberculosis, but tuberculosis evolved a huge resistance against streptomycin. What we think, however, the key is our antimicrobial peptide. The reason why we think that is because the antimicrobial peptide actually targets very generic lipid sequences in the cell membrane. So in order to actually in order to evolve a resistance against the cell membrane, the bacterium would have to create a lethal mutation against a generic lipid sequence within the membrane.
Great talk. My question is about skin commensals. If you're going the topical route, have you taken a look at what this might do to some of the other bacteria that we need to be on the skin to have a healthy microbiota there? Could you be creating dysbiosis?
That is a very good question. And I think we should look to future engineering for that.
Is there expectation that there'd be cross-reactivity with neomycin sensitivity? And has that been evaluated?
We didn't test that, and I don't know. I'm sorry.
Thanks so much. Great talk.
Thank you.